So actually, on last Saturday, we learned the basic structure of neurons or nerve cells, right? And as you can see in the PowerPoints, the neural cells can be divided into three parts: in the cell body, and outside of the cell body, can see the dendrites. Dendrites is the structure like the tree branches. Can see the, like tree branches. And along you know the there is the axon, or in the middle of the neural cell nerve cell, and the axon's function is passes messages away from the cell body to the other neurons, muscles, or glands. But remember, outside of the axon, there one important structure called a myelin sheath. The myelin sheath's function is to cover the axon of some neurons and helps speed neuron impulses. So the keywords here is to cover or protect and speed. You can imagine that there's electricity lines and negative and positive, and actually we have used the the mm, colorful plastic to cover the electricity lines to prevent them to interact with each other. Now also, myelin sheath is like the function of that electricity line, so it can protect them the signal inside along the axon transmission without any interruption by any other neurons. And the end of the neurons cell, you can see that this terminal branches of axons and form junctions with other cells. There are three parts of the neural nerve cells. Nerve cell. Okay. Uh, I remember last time we have mentioned one disease is called multiple sclerosis. I'm not sure you can remember such a kind of disease. Uh, Here you can see multiple sclerosis, briefly MS. That is very frequent diseases for doctors in internal neurology, uh, or if the physician in the department of internal physiology, neurology, they will from time to time um, to see the patients who, for example, cannot see clearly, cannot raise their arms, legs, and to do the in some kind of a movement, precisely. Uh, Generally, such a person, maybe they are very old, in their 70s or 80s, but some of them are in their in a young age, like 20s or 30s. At that time, the doctor will doubt that such a person will got some disease like MS, multiple sclerosis. As we can see from this PowerPoint slide, we can see that because of the myelin sheath of the axon has been destroyed, so the nerve fiber of the axon is exposed, and then it can easily interact with the other fibers. So you can imagine that the signal transmission is not that kind of accurate. So it leads to the diminished or complete loss of neuron functioning in those damaged cells, particularly associated with muscle control and movement. So in the MS, multiple sclerosis patients, they cannot finish the fine or some kind of tiny movement as accurate as they previously can. So that's pity. And the lot of symptoms like include fatigue, changes of vision, balance problems, and numbness, tingling, or muscle weakness in arms or legs. Um, so that's the typical clinical manifestations of the multiple sclerosis. Okay. So I think that we can move on today. Um, we will learn very important concept about how the nerve cell, um for example, why the person feel excited and why the person um feel tired, tired, and then they, for example, I think that you got such kind of like experience that if you climb the mountain, or to be some scenic resort, and then you got to climb a lot, but just maybe 10 or 20 minutes you got to have a break. So why you need a break? Maybe you say, oh, I was tired then. But can I ask you further questions? Why uh, were you tired? And maybe you will think, oh, because I then lack of energy on my muscle cells. Okay, I think that's just one of the reasons. And in the future, we'll talk about the basic structure of the muscle cells. But also, please remember that there is some problems or some mechanism about the nerve cells. Since you can move, it is kind of a cooperation between the nerve cells and the muscle cells. So we firstly talk about the basic structure, the basic mechanism of the nerve cells, how they make the nerve cells themselves excited, and how they cannot excite it themselves. So I would like to share with you one slide here. Let me look for the slide. Let me look for the slide. I'm not sure that we have mentioned this in the last Saturday's class. Okay, here. Uh, I'm not sure that you can remember these slides. This called neural impulses. Impulses. Um, Please remember this picture. This is very important. I make it in a larger scale. Okay, so that we can you know view the picture in a larger way. Uh, I think that you can see they in this x and y axis, the on the x axis it is time 0 1 2 3. Time, the unit of the time is millisecond, second. And on the y axis, the unit or the name is called like the transmembrane potential. Transmembrane potential. Actually, the membrane is the neural nerve cell membrane, right? Transmembrane of nerve cell potential, MV, millivoltage, MV. So you can see, when the nerve cell is very calm, not excited, it is just, I mean, the transmembrane potential is around minus 70 MV. It is called a resting potential, which means the nerve cell then not excited, very calm, peaceful. But with the time last, around maybe the time is more than zero. Okay, for example, you can see zero, between 0 and 1, the time around maybe 0.2, okay, milliseconds. You can see the transmembrane potential has risen a little bit, just a little bit, to minus 60 MV, millivoltage. Please pay attention the word here, threshold. Threshold. Threshold means the minimal requirement to do something. Again, the definition of threshold means the minimal, the least requirement to do something. In this way, the minus 60 MV is the minimal voltage for the nerve cell to be excited, to be excited. So you can see if the transmembrane potential reach the minimal voltage minus 60, it will immediately, immediately rise from minus 60 to almost, you can see the number here, yeah, um, positive 50, positive 50, okay, in a short period of time. You can see around a 1, right? 1 m seconds. Just, maybe less than 1 second, right? You can see because here is more than 0, and here is we can use the, you can see now it's like, it's here it's a 1 millisecond, millisecond. You can see the from minus 60 MV to positive 30 MV, the time is less than 1 millisecond, very quickly. So the process from, the minus 60 MV, which is threshold or the minimal voltage of the transmembrane voltage, potential voltage, rise, quickly to positive 30 MV. This process is called here depolarization. Depolarization. Okay, please remember. Please remember. Depolarization. Okay, depolarization. What is depolarization? You know, when the nerve cell is in a condition of peacefulness, quiet, calm, it is minus 70 MV or minus 60 MV, but it is quickly turned from negative to positive. This, in a polarity has been changed, has been moved from negative to positive. So this process is called a depolarization. Okay, depolarization. Well, the story moves on. You can see after 1 millisecond, there is a process. You can see that the in this part of the voltage would immediately fall down, okay, fall down. So from positive 30 millivoltage to around maybe minus 60, here minus 60. So I think this process is just on the contrary, side of the depolarization, right? So this process is called a repolarization. Repolarization. Re means again, right? Re means again. So then in this process, after this process finished, the polarity of the cell, called a transmembrane potential, right? Transmembrane potential will go back to the original situation, which is minus 60 MV, millivoltage. So this process is called a repolarization. Repolarization. Okay. So you can see from this picture, the voltage, transmembrane potential did not just stop at the minus 60. It is go on, move on, and then to minus 70, and then further, less than minus 70, maybe it's like minus 80 or minus 90 or so, okay. Just imagine here, we can see maybe minus 80. Okay, minus 80. Okay, minus 80 MV. Well, minus EV, minus 80 MV maybe is the lowest transmembrane potential of the neuron cell, but it will not stay more minus 80 MV there for long. It will go back to the potential, the resting potential, which is called I mean the number is minus 70 here, minus 70. So this is the whole story, that the transmembrane potential from minus 70 MV, and like I think the roller coaster, like a roller coaster, climb up and descend, ascend and descend, and then go back to the starting point, 70 minus 70, right? So and then next it will excite it again to minus 60, and then again depolarization, and then when it reach a positive 30, and then it will go down here, and then this process is called a repolarization, and then it will go further, less than 70 minus 70 to reach almost minus 80, and then go back to again to minus 70 MV. So again and again, that is the the repetition of the depolarization and repolarization. Okay, this is very typical, very typical. Okay, but remember, this is talking. We are talking about the nerve cell. Okay, just a nerve cell. Okay, nerve cell. As to the heart cell or muscle cell, or they have their own curves, depolarization and repolarization. Okay, now just remember, this is only they only refer to the nerve cell. Okay, nerve cell. Also, please watch here. I mean, the word clear carefully. Can you say absolute refractory period? Absolute refractory period. So this period, I mean, it's in a like light yellow here, in the absolute refractory. Refractory means cannot change, resist changing. So absolute means 100%, right? 100%. And a period means duration of the time, right? Duration of time. So absolute refractory period means during this period of time, we cannot make it excited further or make excited again. Just please pay attention. This period, absolute refractory period, is from minus 60 MV, and then during experiencing depolarization and repolarization, and when the transmembrane potential reach the minus 60 MV. So this period is called absolute refractory period. So which means if you stimulate the nerve cell, it will have no response, and you cannot make it excited again. So I think if you can reflect on some usual life, for example, you're very excited. For example, you spend the whole night, or maybe you play in your classmates' room, or in some bar or club for long, and you're very excited, and then you go home for sleep. But just imagine your friends or your mom and daddy you want to talk to you or want to call you, and then you will feel very agitated, unhappy, and then by saying that I'm sorry, I'm very tired, I want to sleep, close the door, don't bother me, go get out of my room. Why you got that kind of feeling? Generally speaking, then your you know nerve cell, brain, spinal cord or peripheral nerve cells, all of these cells are a nerve cell, right? So nerve cells, we're talking about the nerve cells, right? Nerve cells. And then after you have excited, which means your nerve cells has been depolarized and repolarized. So you were in the absolute refractory period, and now you don't want to make yourself excited again, you don't want to play more again, you want to what you want is just go to bed, have enjoy your sleep, right? Enjoy your sleep. I think that everybody gets such a life experience, so we can make use of this picture to explain why you have this kind of uh, life experiences. Even though your friends or your mommy or the daddy or anybody you want to play with you or want to talk to you, you don't have the interest. What you want is to sleep, right? What do you want to sleep? Anyway, so after the absolute refractory period, and then I can see the relative refractory period. Relative means to some extent, maybe 2%, 5%, 10%, or 20%, or 30%, blah, blah, blah. That's to say, relative during the relative refractory period, um, it is possible for you, even though you're very tired, and it's possible for you to talk to your classmates, talk to your friends, your mom and daddy, even though you're a little bit tired, but you're not that kind of tired, and you realize that, oh yes, I got some kind of left energy to talk to you. Okay, so you can see in the relative refractory period, we can from that picture can see that, I mean, the time, the duration is like from the minus, I mean, from minus 70 MV and to the next, minus 70 MV, which is called a resting potential. And just imagine if you have, had very sweet, nice dream, nice sleep. So next day, you will feel fresh, energetic again, and then you want to play more, but actually you got to study hard, right? Okay, and you want to uh, do this kind of, for example, you just want to have fun, and then at that time, when you get up in the morning and then get a call from a classmates, hey, buddy, let's go play again this day, today. And then you say, oh, wow, today is a Saturday. Why not play again? And then you feel excited again. You feel energetic. So in that's to say, your nerve cells can be, you know, excited, and then the nerve cells can be depolarized and repolarized again, right? So this is the basic mechanism of the nerve cells, the neuron. Uh, we just make some association of our daily life experience for our better understanding this seemingly complicated depolarization and repolarization. Okay, um, I'm not sure that you can ask some question further. Uh, w- which kind of mechanism lead to the depolarization and the repolarization? Okay. Well, actually, in this picture, in this set of the PowerPoints, we don't have this kind of let me just check. Okay, but first I would like to wipe this my ink. Okay, wipe them away and then make them to the previous condition. Okay, previous condition. uh so the depolarization and repolarizations res- result from the influx and efflux of the sodium ion, potassium ions. Just remember this. Well, the influx of the sodium ion will result depolarization, which means excited of the cell, nerve cell. And if the sodium ion efflux means get out of the nerve cell via the transmembrane, some kind of protein channel, and then since as we know that the polarity of the sodium ion is positive, right? You can just imagine if much more the sodium ion with positive polarity get out of the cell, and then inside of the nerves neuron cell, you can see the polarity will be turned from positive to negative. Now here, minus 70 is negative millivoltage, and then it will be repolarized. So depolarized, you just remember, depolarized means the influx of the sodium ion. Repolarization, generally, if we just refer to the sodium ions, and then the sodium ion will get out of or efflux from the internal of the neuron to the outside of the neuron, external. Okay, which means briefly called efflux, influx and efflux. Okay, so that's the I mean called the basic mechanism of the polarization and repolarization. If we just make use of the so-called polarization, depolarization, repolarization as the basic mechanism to illustrate to the nerve cells excitement and refractory periods, etc. Okay. Anyway, I don't think that this kind of illustrations or is easy for you to understand. A little bit difficult to understand if you previously don't know what's going on about the neural impulses. Anyway, um, it's a little bit basic. It's very basic, and a little bit challenging to remember this, but no problem. And no problem. I just give you some kind of memorize way to associate with your daily life. You can see when you're excited, you want to do anything. When you are tired, you don't want to do anything. You just wanted to sleep, enjoy the sleep. That's why we call it the absolute refractory period and a relatively refractory period. Depends on your neural cell situation. Okay, neural situation. Anyway, uh I think that is the oh, how about we just yeah uh this is called depolarization and repolarization, and then here you see the outside the axon is called a positive ion, and the inside the axon is a negative ion. Okay, that this is called the general situation, called not firing. Here not firing means not excited, or in the resting potential, minus 70 MV. Okay, if they're firing, and then which means they excited, and then the positive ion, which is I mentioned positive sodium ion will get in, right? And then the polarization will be changed from negative to positive. This process is called depolarization, right? Depolarization. Anyway, so in this picture, we can see the neural communication. So this arrow here, this arrow, you can see this arrow means the sodium ion get into the inside of the neuron, and this means the uh, sodium will get out of the neuron nerve cell. And again, to the next part of the axon, the sodium ion will get into the nerve cell again. And here they get out, and then to the next part they will get into, get out, and get into, get out, and get into. So this is the basic mechanism of the neural communication, or how the signal, the nerve signal, can be transferred toward the axon terminals or direction of neuron impulses. Okay, neuron impulses. Um, I remember last time we have mentioned this. Um, the nerve cell, if the signal has come to the nerve ending, uh, and how the signal transmitted from one nerve cell to another. There is one important structure called synapse. Here, this is called synapse. Synaptic cleft or synaptic cleft, synapse. So synapse cleft got two membranes called presynaptic membrane and postsynaptic membrane, right? Pre means before, post means after. Okay, so you can see if we just enlarge, here, this small part in a larger way, you can see on the presynaptic membrane, there's a synaptic vesicle, and inside the vesicle, there are neurotransmitter molecules, okay? And then the neurotransmitter molecules will be released to the synaptic cleft. And the neurotransmitter molecules will, you can see, you can imagine they will swim to the postsynaptic membrane. And it's very lucky they're on the postsynaptic membrane, they're like receptors, or you can see binding sites. Here, the binding site, the binding or receptor is actually is kind of protein. Okay, it's kind of protein, and then this kind of the neurotransmitter molecules will bind the receptors on the binding site. For example, you can see in the middle, this is called a sodium channel. Sodium channel, and then the sodium channel here at first, the binding site, you can see there is some kind of the door is not 100% or completely open, but in the middle the channel has been 100% open, because or after the neurotransmitter nucleus binding to the binding site. So in this way, the sodium ion with positive electricity, with positive, ion and sodium will get into the post membrane, postsynaptic membrane, right? And then you can imagine, thousands, millions of this sodium ion with positive, electricity or polarity would get into the next nerve cell via the channel, right? As we can, as we have told that the influx of the sodium ion means the depolarization, right? Depolarization actually, to some to some extent is equivalent to you know uh, excitement, right? Depolarization means excitement. So you can see more and more sodium ions get into the next in a nerve cell, and in this way the signal of the nerve cell of the previous nerve cell will transmit the signal to the next nerve cell. That is the general or basic process of neural communication. Okay, so up to now, actually, we have talked about the two, uh, parts of the nerve cells, mechanism. One is in the one nerve cell, how the nerve cell, excited themselves and how to, make themselves come back to the, peaceful situation and come to the resting potential, which is minus 70 MV as the transmembrane potential. And second part, we talk about how the nerve signal can be transmitted from one nerve cell to another. Okay, I think this picture, the picture we're sharing, the slides are very important. Okay, I've finished presenting this, the neuron communication. But please be noted, we will talk about the neuron transmitter here. Neuron transmitter. Neurotransmitter actually is kind of hormone, or some, here can see some neurotransmitter and their functions. Okay, uh you just remember there are two kinds of neurotransmitters. One is positive or excited, or another is kind of inhibitory. Okay, inhibitory neurotransmitter. um for your better memory, I think first please remember GABA. Okay, please remember GABA. GABA. Okay, GABA. GABA's full name is gamma aminobutyric acid, which is a major inhibitor neurotransmitter. Uh, so for example, for example, you tired, you want to sleep. uh it's good, right? Because without sleep, and then you cannot regain your energy, right, for the next day. But just imagine one patient who cannot fall into sleep, for maybe any possible reason. Okay, I don't know. Maybe the person who, for example, insomnia, cannot get into sleep, will complain, "Doctor, help me. How can you, help me have a very, nice sleep?" Uh, as the nerve doctor, maybe the person will think about the neurotransmitter, like GABA, inhibitor neurotransmitter. For example, if the person's GABA secretion, not that kind of adequate, the person on the contrary will be excited, right? Will be excited. And then you can imagine that the person, excited, that's to say he or she cannot fall into sleep, right? Cannot fall into sleep. So GABA, if undersupply, linked to seizures, tremors, and insomnia, right? Insomnia means sleepless, right? The tremor means shake a lot, shake too much, your muscle shake too much, your body shake too much. Seizure is a kind of disease that the person cannot, generally it is the muscle will be overexcited, and or the person, if the person lost his consciousness, and then maybe he will, bite his or her own tongue, or even fall into the, in the ground. I think that's overexcited. I mean, the muscles are overexcited. So that's one of the, mechanism of the GABA. So even though it is inhibitory, but sometimes we need it, especially you want to, for example, you need the sleep in the evening, right? So GABA is the only, okay, inhibitory neurotransmitter for us to remember. Uh, in this table, you can see one, two, three, four, five, six. There are six kinds of neurotransmitters. The rest five neurotransmitters, generally speaking, are excitatory neurotransmitters. Let's just say they can, help you to keep excitement. Okay. Uh, the first one is, acetylcholine, briefly ACH. Okay, briefly ACH. It enable muscle action, learning, and memory. If it is, in malfunction, and then maybe the person will go to Alzheimer disease, or ACH producing neurons deteriorate. Uh, I think that you know what is Alzheimer disease, right? So Alzheimer disease is the uh, aging people with the tendency of stupidity, right? Stupid, you know, lost, you know, lost memory, lost the smartness. In the end, just like uh, uh, cannot remember anything in the past. Okay, it's it's in a point of view of the neurotransmitter, maybe the ACH acetylcholine secretion is not adequate because the ACH acetylcholine is associated with memory, right? Memory, associated memory. So I think that maybe you will probably come across some commercial or advertisements on the TV or via the internet. Hey, you want to improve your memory ability? I got some kind of secret agents. You want to try? You want to try? Well, I think that to some extent or in a high probability, this so-called secret mysterious agent must be, acetylcholine. Okay, but I got to tell you that even though in the laboratory experiment, the external, ACH, it maybe it is, it works for some animals, but if you just input, inject some external ACH to your body, like the IV transfusion, we up to now we have not get any positive result to improve the memory. It, you know, even though this kind of the commercial is very popular. Okay, so maybe it's kind of like a placebo effect. Placebo effect means maybe it is useless, but because you think it is useful, and then it is useful. It's the effect of the uh you know your thinking is like a psychological phenomenon. Okay, so next is dopamine. Okay, dopamine. So I think the dopamine can influence movement, learning, and attention, emotions, right? um uh I think that when you love something, like somebody, and then your secretion of dopamine will, in a relatively high concentration, but will not last long, right? So excessive dopamine receptor activity is linked to schizophrenia. I'm not sure that you know this word schizophrenia. Schizophrenia, schiza, actually schiza, SCHIZO. It's a German, means separate, split or separate. Heophrenia means kind of the spirit condition, or schizophrenia in English, in plain English, means splitted uh you know mind, split mind. Okay, so for example, the person got the schizophrenia, and then if you ask him, hey, what day what day is today, generally you will think, oh, today is Saturday. Or what's the weather today? Oh, today is sunny day, blah blah blah, right? It's very reasonable, very normal. But for the patients with the schizophrenia, they will not think in that way. Will think, oh, what day is today? Well, I like your weather. I like your clothes. I like your style. But clothes, blah blah blah. But actually, clothes is different from your question. What day is today, right? So this is means the person with the schizophrenia can think, too actively, and then they will think from A to B to C to D. But generally, we just ask you from A to B, and then so I mean, their thinking are too excited. So you can see the stop of the dopamine, the brain produces the tremors and decreased the mobility of Parkinson's disease. So Parkinson disease, I think that because the tremor of the hands, the person that got a Parkinson, and it's also usually happening on the aging people, right? Aging people. So dopamine is associated if lack of the dopamine secretion, it is associated with the Parkinson disease. Okay. Serotonin. Okay. Serotonin. We can briefly write serotonin into 5-HT, 5 hydrogen, 5 hydroxy tryptophan, or briefly 5-HT. Okay. So serotonin is also excitatory neurotransmitter, affects mood, hunger, sleep, and arousal. Okay. But please remember the lack of the uh, you know secretion of serotonin, the person possibly will get the disease called depression. Depression. Okay. So if we just want to treat the depression patient with some agent, generally in America or around the world, the agent called Prozac. The Prozac is the serotonin receptor inhibitor, reuptake inhibitor. If we just inhibit the reuptake of serotonin in our blood, that's to say, the serotonin will maintain relatively high concentration in our blood. So the serotonin can move along the blood to the rest of the body, to some nerve cells, and then make our the depressed patient excited or a little bit excited. So that is the mechanism uh, of the doctors how to treat the depressed patient. Okay, just remember that serotonin is associated with the depression. Okay, and there's norepinephrine. Norepinephrine or epinephrine generally control the alertness and arousal. You just remember one short expression called fight and flight. Okay, fight means you want to fight against your enemy, or opponent, and then flight, you wanna means you wanna run away, right? No matter fight or flight, you gotta make your spirit arousal, and you keep your body, keep yourself alerted, right? Alert. And so just imagine if the lack of the secretion of the norepinephrine, and then it will lead to depressed mood, not high, not feel high, right? Okay. And the last, the glutamate. Okay, glutamate actually is some uh major excitatory neurotransmitter, and then they involved in memory. um Also, in our daily life, there's a glutamate in our called the condiments. If you want to, for example, if I think you think, oh, this dish is very yummy, tasty, and then maybe there's some condiments which is called the glutamate, has been added into the dish, because the glutamate is kind of excitatory neurotransmitter, and then it will know be put into your mouth along the dish, and then there is nerve cells, nerve ending along your tongue, your mouth, and then you will immediately sense the oh, it is very yummy, because the glutamate can make you feel excitatory. So that's the the explanation why you think the food is yummy. But if the oversupply of the glutamate can overstimulate brain, producing migraines or seizures, which is why some people avoid MSH. You know, if the person feel a little bit of headache, that here migraine means the you feel headache on half of the brain, maybe left, maybe right. Okay. um why the person feel migraine or migraine? One explanation is that because the glutamate has been oversupplied and overexcited, and then the migraine occurs. Okay. So for, if you can see, if a person has used his or her hay brand for long, and then the brand itself send some alert, alarming signal to the person, hey buddy, you need a break, you go to bed, have a break, have sleep. So generally, the migraine patients will feel better after a nice sleep, maybe next morning, and then you ask him or her, hey, how about your headache, how about your migraine? The patient said, oh yeah, I feel bad, I feel good. So let's keep playing, keep working. So you can see, because during the sleep, during sleep, the glutamate secretion will be, lured. But on the contrary, the GABA here, GABA, gamma aminobutyric acid, will be secreted, make the person not that kind of excited, right? So the migraine patient saved. Okay. So this is, the general introduction of the neurotransmitters and their functions. It is very basic, uh, but very important. Very important. Okay. Well, uh, the time almost to come to the end of this today's session. If you have questions, feel free to ask me. Okay. I think that question and answers, this kind of QA is very important. Have any question?
Could you wait, sorry, could you tell me what MSG meant again?
Oh, the internet connection is not that good. Would you please, repeat your question, please?
Oh, okay. I just wanted to ask, what does MSG mean?
Oh, avoid, you know, MSH mean is kind of like some people avoid it. Here let me confirm. So oversupply, overestimated membrane producing migraine or seizure, which is why some people. Oh, MSG is kind of agent which will stimulate the secretion of glutamate. Okay, it's kind of a excitatory or stimulatory agent. Okay, it's kind of chemical agent. Okay.
Oh, okay. So is that similar to like coffee?
I'm not sure. I'm not sure. I don't want to mislead you, but I just can see that this MSG, uh, if it MSG, I think it will excite it, and then will secrete the glutamate, and then maybe the person will got some migraine headache. Okay. Well, I have found the full name of MSG. Okay, MSG. Here MSG. I will send you via the WeChat system. Okay, MSG. Okay, equals. Uh, can you see the
Yeah, I
yeah,
can see.
yeah, MSH called
Oh, okay.
monosodium glutamate. G here, glutamate, right? Glutamate. Okay, MSH monosodium. Sodium means Na, right? And sodium Na, glutamate. So actually, MSH means the glutamate itself. Okay, so in this slide, we can see that the people avoid which in I can ask you which kind of people should avoid MS or monosodium glutamate?
Um, like people who work too much,
Yeah,
maybe.
yeah, yeah, sure. I think that firstly the person got some genetic problem of the headache or migraine, right? You can see, oh yeah, if you think, oh, I feel migraine from time to time, and I need a should the person recommended to use some glutamate or MSG? No, I don't think so, right? Anyway, any question?
Yeah, I think that's it. Thank you.
Oh, you're welcome, CC. You're welcome. Okay, well, today we talk about I think if I were the student, I feel a little bit complicated because we just mentioned the basic mechanism of one cell in the signal nerve signal transmission inside the nerve cell, and then also mentioned how the signal has been transmitted from one cell to another. Okay, and we mentioned the neurotransmitter, their functions. Okay, two kinds. One is inhibitory, GABA. The five other excitatory uh, neurotransmitters. Okay. Um. Anyway, you will if you want to learn the biology or physiology in a better way, pay some time and attention on this kind of stuff. But I suggest you to associate with this kind of knowledge with our daily life, and you will feel it's very interesting and easier for remember, right? Okay. Well, so that's all for today's the class how the body works. So that's all for today's session class. Thank you for joining me. So looking forward to meet you next Saturday. Have a nice weekend, CC. Bye bye.
Thank you. I'm looking forward to your class next time. Thank
Yeah,
you
bye
so
bye.
much.
Yeah, bye bye.
Bye.